Okay, we just finished up the light-dependent reaction, so let's move on to the second part, the light-independent reactions, also called the Calvin cycle. The purpose here in the light-independent reactions, as you can see the title on the worksheet, is to produce sugar. Specifically, what we're trying to produce is some glucose. So what does the Calvin cycle use to produce these sugars? There's three things that we went over that are used in the production of these sugars. Two of those were produced by the light-dependent reactions. That's why that has to occur first. And those two things are the ATP and NADPH are used for energy. Along with the gas that plants have to take in because they need some carbon. So that is CO2, carbon dioxide. So those are the three things here that are needed during the Calvin cycle. Again, the ATP and the NADPH come from the light-dependent reactions and are needed for energy, and that energy is used to drive the Calvin cycle. And then we need that carbon dioxide because we need the carbon. Why are they called light-independent reactions? Because sunlight is not used here is the obvious answer. The energy is provided again from the ATP and the NADH. So again, the ATP and the NADPH provide the energy instead of sunlight. What makes it a cycle? In any cycle, you start or you end with what you start with. In this case, that substance that we end and start with is called RUBP. So you start and end with RUBP, ribulose biphosphate. Okay, so again, it is a cycle, and you can see that cycle pictured here right in 13. In these particular blanks, I know it was a little confusing for some. I had some questions asked of me, and you had to insert some boxes here, so I'm going to go. But what it was wanting you to do is count the carbons that come through here. Okay, so what is going to happen here if I can insert some text box here? I got to do a little conversion here. Now I can insert my text box. I'll put it right here for a second. Let me format it so everything is right. I don't want a line. I don't want any fill. And then I want it to be bold and red. Again, we are going to start with RUBP. Okay, we're going to start with RUBP, and RUBP happens to be, let me do it this way, happens to be 4 carbons. Okay, so it happens to be 4 carbons. Okay, so we're going to start with this 4th carbon compound. Again, this is your RUBP. Okay, and we're going to join a carbon here. And what we end up with over here is, let me copy this and paste. What we do is we end up over here with something called 3-phosphoglycerate. 3-phosphoglycerate only has 3 carbons. Okay, now you can see they are keeping numbers on the way, so I should have been doing that also. So let me go back here. We're actually going to start with 6 of these. Okay, and we join that with 6 carbon dioxides, and we actually end up with 12 of these phosphoglycerates as we move around. So we have 12 ATPs. Again, the ATPs and the NADHs again are providing us with energy. Okay, and when that happens, we end up breaking off a phosphate group. So what we get out of it is ADP. Okay, once that phosphate gets broke off, that gives us energy. Same thing down here with NADPH. Okay, what is going to happen is the hydrogen gets broken off, so we end up with NADP+. Sorry about that, need to stretch this out a little bit. So we again end up with NAD+. Okay, now that energy is used to convert phosphoglycerate into a substance known as G3P. Okay, G3P. Now, as this G3P continues, and you can see there are 12 of them, two of them actually are going to go here. So they didn't have you label this, but I just want to move this down here so you understand that 2 of them go right here. So this is your G3P. The other or remaining 10 cycle themselves back around. Okay, so of these 12 that produce, 2 are going to go into making sugars. These 2 right here. And 10 of them are going to go into remaking the RUBP. Okay, now this does require some energy. And you can see that you have 6 ATP coming in. So we're going to end up with 6 ADPs coming out. Okay, so here we have, again, we're starting with our carbon fixation up here. This would be our regeneration. This is our generation part, our regeneration part, and this is the conversion of the phosphoglycerate into G3P. So step 1, step 2, step 3, step 4. Okay, those are your 4 steps in the Calvin cycle. So again, I just want to reiterate here. Okay, so we're going from here to here. Again, this is a process called carbon fixation. So these are the steps that I gave you. And then that carbon fixation is what is going to produce over here our phosphoglycerate. Next, we have our reduction. During reduction is when the phosphoglycerate gets converted into our G3P. From there, we go to generation. Actually, I'm going to move this part to this line right here. We are generating our glucose or our sugar. And then finally, some of that goes into regeneration, or the reproduction of our UBP, so our cycle continues. So carbon fixation, reduction, generation, as we see right there, and regeneration. Okay, there's again your 4 steps. So let's move down here. We talked about several factors that are going to affect the rate at which photosynthesis occurs. I ask you to give 3 here, so we can just list these out. Okay, one of those we went over was temperature. Not very good spelling. I'm just going to list a second one on this line. We talked about light intensity. Obviously, more light is going to allow it to be better. A third one that we mentioned, or that we did mention, was the availability. And I am struggling right now. How about if of water. Also, you could throw in here, and you maybe have had this, the availability of CO2. As a factor also, because obviously the amount of water, you need water, we need carbon dioxide, we need energy. So those 3, obviously, since those 3 things are required. And then we talk about temperature because there are special enzymes at work here. Okay, and if it gets too hot, those enzymes begin to break down. However, if you don't have enough light, which is producing the temperature, you're going to be low. So when temperatures get too low, when it gets too cold, this is when the chlorophyll starts to break down. That's one of the reasons that plants shed their leaves and everything during the winter. And they do not photosynthesize anymore, but they rely on the stored sugars that they have made when it is warmer. If you place an atmosphere of pure oxygen, would it be able to carry out photosynthesis? And the obvious answer here has to be, if you thought about it, a big no. Why? Because CO2 is needed for photosynthesis. Okay, CO2 is needed for photosynthesis. So obviously, if you do not have any CO2, if you have nothing but pure oxygen, the plants are not going to be able to photosynthesize. And then I did not talk about this, but I did ask you guys to read over this. So very quickly, we're going to go through here. And the first one you guys should have read was called C4 photosynthesis. And this occurs in plants that have specialized chemical things. Examples of these are corn, sugarcane. It would be another example of that. And get that tamed and get that centered. And can't seem to get that centered to where I want it. There we go. And then we also have pineapple trees, desert cacti, and ice plants. Okay, what's this called? They use something called the CAM method. CAM. And what happens here is that these plants can, these plants can allow CO2 in O2 in at night, and because their this is allowing this to occur at night, this minimizes the loss of water. So again, this is why, again, how about if I learn to spell a little bit?